My guest today, Dr. Thomas Hartung. You, sir, if I am correct, are the Doornkamp Zbinden Chair. Am I pronouncing that even close to correct?
Actually, pretty good.
Okay, thank you. You're very kind. For evidence-based toxicology in the Department of Environmental Health and Engineering at Johns Hopkins, Bloomberg School of Public Health. You have a joint appointment at the Whiting School of Engineering in Baltimore. And you also hold a joint appointment for molecular microbiome and immunology at the Bloomberg School. You're an adjunct affiliate professor at Georgetown, Washington. Very good. In addition, you hold a joint appointment as a professor of pharmacology and toxicology at the University of Constance in Germany. You're the director of the Center of Alternatives to Animal Testing, a collaboration between Johns Hopkins University and the University of Constance. You are field chief editor of Frontiers in Artificial Intelligence, and you've authored more than 675 scientific publications with more than 47,000 citations. Your toxicology classes on Coursera had more than 19,000 active learners, and you steer the revolution in toxicology to move away from a 50-plus-year-old animal test to organoid cultures that use artificial intelligence. Quite impressive, sir. Your research interests include toxicology, pharmacology, infectious disease, alternatives to animal testing, microphysiological systems, cell culture, validation, pyrogen testing, big data, artificial intelligence, metabolomics, developmental neurotoxicity, and mini brains. Interesting. Let's start there. What are mini brains?
And we have already filled the first 5 minutes of this interview with these accolades. No, that's the problem of getting older and older. You accumulate titles, and
Exactly.
But I'd have to say that to some extent I'm proud of this because when I started in the field of alternatives to animal testing, some people, German Research Council, and so they said, "How can you risk your wonderful career by going to such a niche area?"
Right.
And now have a chair at one of the most prestigious universities in the world, and many other appellations. My life, I have got some lifetime achievement awards, which is really scaring, especially from the very organization which told me not to go in this direction.
Oh, the irony, sir.
Yeah, okay. No, but this is fun aside. You see from all of this that I'm broad, not deep. Yeah, there's some scientists who find a niche or some area and go deeper and deeper.
Yeah.
And they're the pope in this field at some point. I'm trying to connect different areas and create movement between these. Yeah.
Right. Right. Now, your affiliated organizations, how much time do you spend at each? Are you located in one particular place and work constantly from there? Or do you move around a bit?
I mean, travel comes with the territory. I have to deal with this, but this is more to conferences. I'm located with my family in Baltimore. My wife is professor too, and we work as a team. And Georgetown is just a bit more than an hour south of Baltimore. Constance is a bit more challenging on the other side of the Atlantic, but I've been there for 35 years, did many studies. And if you know each other, then Zoom is a godsend. You can really deal with many things without physically meeting.
For sure. Now, you give a lecture online that I checked out in which you talk about no more lab rats. Our shared friend and colleague, Dr. Charu Chandrasekhara, she's been on the podcast, and her work focuses research away from the use of lab animals. Can you tell us about your efforts in this regard? How long have you been working in this area, and towards what end?
Yeah, I would say it's 35 to 40 years now. I'm already in my study years. I started to work on cell culture experiments, master's thesis, PhD. All of this was around cellular models in the end to replace animal testing. In the beginning, it was really for me more using the most modern tools in order to model this. Charu is a dear friend, and she's directing a similar center in Canada. But her background is more on the animal welfare advocacy side. So that's her angle. I come really out of science. Yeah, I'm a scientist, and I'm trying to use sound science and also the credibility which comes with really competing for money with other researchers, putting paper out which are in renowned journals. So much more of this to reach this audience. You know, you need to be a scientist yourself to be listened to by other scientists.
Mm-hmm.
And this is why I'm really taking all of these roads at
Mm-hmm.
the same time to stay credible with this audience, who very often have already made up their mind. Yeah, they like or don't like animal experiments. They use them or they don't use them, and it's very difficult to shift one of them.
Mm.
Yeah, they occasionally follow new opportunities, but it's very difficult to tell them it's not good what you have been doing.
Right.
It takes a lot to turn somebody against the methods they have been using for a long time.
And is that simply because animal models have been used for so long, it's just become so ingrained and entrenched in the fields that people are not familiar with alternatives? You mentioned cell cultures and things like that. Why are they slow to make this realization?
I think that we use what we have at hand.
Mm.
Lab rats and mice have been used only since the 20s of the last century, so quite exactly 100 years.
Right.
Science did not really rely on any of these regularly and in large numbers. Yeah,
Mm.
and it for many people it seemed odd at the time that they could reflect humans. Yeah,
Yeah.
then it was simply convincing how fast you could produce findings in the life sciences. And then there was a long period until other methods really came about, and cell culture started somewhere in the 60s. I mean, you can always cite very early attempts, but really as a more regular tool, I would say it was really only in the 80s of the last century that we got cell culture, and I would say computers are to really replace some animal testing. That's a very recent thing of this century.
Mm-hmm.
You can always cite some examples. Yeah, somebody did already something, but we are now having the tools to really replace.
Good.
And then it is this generational change. Yeah.
Mm-hmm.
Thomas Kuhn has written as a philosopher. Yeah,
Oh,
he wrote
I'm familiar with Kuhn. Yeah.
his Nature of Scientific Revolutions is a very, very eye-opening book from 1970, and he made clear it is not a continuous change in science, but it is in waves, and these waves are very often coinciding with the retirement of a chair of a
Mm-hmm.
department. Yeah, people are not changing what they are doing.
Right. So based on Kuhnian philosophy, then would you maintain that what you're doing is a paradigm shift in scientific research, that the old way of doing things is no longer the way we need to do things?
Yeah, absolutely. I think my own main lesson, which I took and try to share, is animal experiments are terribly overrated or in what they can actually deliver. Yeah?
Right.
I was very surprised in 2008. I wrote a review which was trying to summarize how bad or how good animal experiments are in reflecting human.
Mm-hmm.
And I was surprised that with millions of articles in the scientific literature, I did not find a single larger review paper which was attempting just the same. In the meantime, there are some. Yeah? And I'm collecting data since, and what I can see is mice and rats predict each other at about 60% of the time for anything which is a bit more complex, like cancer or effects on embryos. And there's no reason to assume that either of them is better in predicting humans. On the contrary, yeah,
Mm-hmm.
they're obviously much closer. So I think this is a this was for me an eye-opener and also changed the entire argument. It is good to be on the right side of ethics and avoid suffering of animals where not necessary, but it is also about better science and better decision taking. Yeah, and this is my selling point. Yeah, I'm not going here. Oh, these poor rats, and it is really about can we help product development, especially drug development, to make it more efficient, and this almost automatically leads to look out for new methods.
Yeah. So if I'm hearing you right, you're saying even if we take compassion for animal suffering out of the equation entirely, it's just a bad model. It's just not an efficacious model for producing the types of results we would like. So it's you're stating a scientific fact.
And I can back this with many studies.
Right.
Whenever this has been analyzed a bit more systematically, and we have done so for several thousand chemicals, which have been done in very standardized tests. And this is the advantage of toxicology. In toxicology, we use the same protocols for many decades. And the people are extremely well trained. There's internationally binding guidelines how to do it. So we really have thousands of examples where the same substance has been tested several times. Or we have tons of studies where we also know what happened then in human clinical trials after these animal studies had been passed, if you look into drug development.
Yeah, right.
So it is toxicology is a small field. Yeah, it is 10% of animal use, roughly.
Oh, okay.
But it is has a lighthouse function because here we really have the data. There's no reason to assume that animal experiments are any better in the other areas where we simply cannot compare as easily.
Right. Interesting. Now, you co-authored a paper called Artificial Intelligence. It's the end of the talks as we know it, and I feel fine. Aside from the nice reference to REM, one of my favorite bands as an undergrad, what can you tell us about your findings?
Yeah, I mean, this is REM is also one of my favorites. Losing my religion is my absolute favorite, but that's a different story. Yeah. In this case, I'm losing some of the old beliefs, at least in toxicology.
Yeah.
That's why it is also fitting. But I think the important thing, Nicole Kleinstreuer, my co-author, and me, was we were summarizing in this quite extensive paper how the advent of artificial intelligence at the moment is changing everything, and including the safety sciences. But here we can again demonstrate how advantageous this is. I mean, everybody sees this at the moment. The last 2 years were incredible with respect to generative AI and these things. But the first example I can give you what is changing. It is AI is allowing us to find whatever is in the public domain about a certain thing.
Right.
And this is a tremendous problem. Yeah. In science, a lot of information is at very, very strange places, and it's very difficult to find it all. Sometimes it is astonishing much. If you take bisphenol A, for example, there's more than 12,000 papers written on bisphenol A.
Wow.
So here it is not the problem to find anything, but it's the problem to find the relevant thing among all of the noise. Yeah? Again, something where AI can actually shine. And a year ago exactly, I was asked by the EPA to peer review a risk assessment, which means they had a substance which had little data but was relevant in certain settings. And they said, we have put 2 people for 1.5 years on this to find all the information they could identify for this substance. Yeah? Then they took some conclusions on this. What does it mean about the risk assessment? After I did my peer review that they did a good job, for the fun of it, I used some of these large language models.
Yeah.
I played around about an hour using 5-6 versions to see also the improvement over time. I found all of the information which they had identified. And I even found a complete risk assessment, which was carried out by another institution, which they had completely missed.
Gee.
Yeah. So you see how efficient alone getting you on a silver platter information is, as long as it is in the public domain. But this was also necessary for the EPA colleagues. They would not have found anything which is behind paywalls or behind firewalls of a company. So they were on the lookout for exactly the same type of information.
Right. Right.
And then the next step is obviously evidence integration.
Mm-hmm.
And humans are not very good in this. Yeah. So if you think there's a lot of different studies which are very different in what they measured exactly, it's difficult for us to interpret this.
Mm-hmm.
And AI shines here. AI is really works. The bigger the data, the more diverse the data are, because it is training from all of the different possibilities of mixing information and optimizing a process, and can really help us to make predictions. They're not yet good enough that you can say we don't need a risk assessor anymore, we don't need an agency,
Right.
but they are helping. I call this a co-pilot. Yeah. So the
Mm.
human must be the pilot. And also look, is there hallucination? Does it make sense? Yeah, there need to be plausibility checks. It is more an alert system, but it is helping you to be much more efficient in this sense. Yeah.
Oh, very good. Very good. Um, Now, a colleague of mine once said, if it takes the lives of 1 million rats to save one human life, it's worth it. Do researchers such as yourself ever think in quantitative terms of animal versus human suffering in this regard? So, is there a point at which we can use animal models or should use them in certain cases, or are they entirely irrelevant at this point?
They're definitely not irrelevant.
Okay.
And a lot of the things we know came from animal experiments.
Okay.
Many medicines were developed on the basis of animal experiments because they were developed before we had anything else.
Right.
And then it takes to um, fill in more and more of these new test systems, these new approaches, and build trust into them. If you take a decision, I mean, fortunately, we never have this decision to take how many humans do we kill in
Mm-hmm.
order, and how many rats are against it. It's all about, let's say, is it economically leading us to good decisions?
Yes.
Something where we can have enough evidence that we can tell a company after spend 200 million to develop a new pesticide or 2 billion to develop a new drug, you don't go to the market,
Mm.
which is a terrible decision. Yeah, this can kill the company. Yeah.
Yeah.
So for this reason, you have responsibility. I'm involved at the moment in an interesting project about the ethics of regulation, because it is on the one hand, yeah, to protect this one child, yeah,
Mm-hmm.
but it's also about keep an industry moving. Somehow, um, you know, a perfectly safe world
Mm-hmm.
would possibly be one without any chemicals. But we can also show that we have tripled life expectancy since modern chemical industry has started. You can show that drugs, despite their side effects, are tremendously important to prolong lives. And it is extremely difficult to say how much negative, how much risk are we willing to take, how much delay do we want to experience,
Yeah.
and then not treat people who also die from this. Yeah, so it is an extremely difficult decision, and it is one where the decision taker, the risk manager,
Mm-hmm.
um, needs to be permanently also trained and updated. And this is why we are trying, for example, to train the regulators, train them in what these new methods do, so that they can decide, can say, yeah, this is what it's giving me, and this is where I not yet can rely on it, or I combine. So this is change management,
Mm-hmm.
and this there we are again with Thomas Kuhn. It is something which is a paradigm shift, really,
Yeah.
when introducing these new methodologies.
Right. Can you walk us through um, a day in the life of Thomas Hartung, like when you go into the lab and what are the types of things you do? Because when I hear toxicology and animals, I'm thinking of rabbits getting stuff sprayed in their eyes. You know what I mean? To determine if a perfume is safe, or that's the classic type of public conception of toxicology research. What really goes on in your lab? What how do you and how does it relate to new models of testing?
Yeah. I mean, first of all, I'm working with a team.
Hmm.
So in Baltimore alone, we're 30, 35 people at any given time at the moment, and I'm not the one in the lab anymore.
Mm.
Yeah, the contrary, when I put on a lab coat, they know I'm either coming with a journalist who wants to take photographs, or they shout, "What are you doing here? Don't disturb our work." Yeah.
Right.
I also have to say that I have been embracing more and more the AI part.
Okay.
So I'm working with a lot of computational methods because I think that this is really a disruptive technology.
Yeah.
Over the last 6 years, I've been the founding editor of Frontiers in Artificial Intelligence, a journal which has now published 1,000 papers
Mm.
this year, John. So it's really becoming a player and a recognized player. Yeah.
Hmm.
So and a lot of my work is actually communication in writing, in endless Zoom meetings, but also tons of presentations. Um, Today I was speaking at a symposium in Rome. I was supposed to go there in person, but then had to change to virtual. But last year I did 88 of these talks. Yeah.
Wow.
17 of them were keynote lectures at conferences, which means the big plenary talks where they use the old farts to make an impression on the audience. But still, it shows you a bit the recognition. And I'm really a science communicator now. Um, my You mentioned that my papers are heavily cited
Hmm.
in this introduction. An average scientific paper is cited twice, once by the own group and once by another group. That's the average. And if my close to 700 papers are now cited 48,000 times, you can see that I have a much better outreach
Hmm.
recognition. Yeah.
Yeah.
Citation is something you can really, which is helpful as a measure of impact, because I only cite what I think is a point of reference for my work, or which I have to acknowledge because it was inspiring for my work. And for this reason, I think this is a very important performance indicator for myself.
Hmm.
And I'm spending a lot of time on writing articles and trying to communicate ideas. Um, you cannot write always the same stuff. You need to really digest things newly, package them newly,
Yeah.
and that's a lot of my business. Yeah.
Yeah. Yeah. For sure. Now, as a philosopher, I'm always interested in theoretical foundations of like human ethical behavior. Are there any particular ethical principles or theories or precepts that guide you in your desire to reduce animal suffering? So I don't know if you can see down here, we've got Peter Singer's book, Animal Liberation. Peter Singer was on the podcast, and he's a utilitarian, right? So the greatest good for the greatest number. Do you have a particular school of thought in philosophy and ethics that you follow?
I mean, I'm not a philosopher, but
Mhm.
I'm trying to be informed about this. So I also met Peter Singer several times over the last 2 years. We are at the moment writing jointly an article about AI replacing animal testing and its opportunities. And so I really like these things. And Hans Jonas and his responsibility ethics was something I think it's a bit blue-eyed, but I liked it a lot. I have to say that I also I like Sloterdijk as a German philosopher. He has written a lot about the critique of cynical reasoning is one of his major works. And
Mhm.
what he introduced is that we're doing a lot of things despite knowing better. And I think this
Yes.
describes very nicely how we stick with animal tests despite knowing better. So I'm at the moment packaging some of these things actually into an article, because I think it's sometimes good to go back to these basics. And there's at the moment a discussion on the level of OECD, so the Organization for Economic Collaboration, who's setting the standard on how to validate a new method against the old ones.
Mm.
I'm trying to influence this with a few articles, and one of them is really basic. Yeah? And this Canadian project on the ethics of regulation was an important stimulus. Or we have an exciting project which is called Organ Intelligence, where we are trying to make brain models, little tiny human brain representatives,
Yeah.
make them learn and memorize. We call this organ intelligence because we use AI to communicate with the system. But you can imagine how many ethical questions there are if suddenly a little brain is becoming self-aware, at least understanding where it is, what is expected from it, and when do you have to stop? Yeah.
Yeah.
When would there be a moment where you can no longer stop feeding it because you have a responsibility?
Yeah.
Or the question, how do you have to inform people who are donating their cells to build these type of things? And they're expecting I'm doing traditional type of cell culture. Yeah? They might want to be informed differently if it is about these type of new methodologies.
Mm-hmm.
I mean, to be clear, we are very far away from anything which is meaningful here.
Mm-hmm.
Our models have about 50,000 brain cells each, yeah, which is about as much as a housefly, yeah. So
Yes.
even if these are human cells, don't expect them suddenly to talk to you, yeah.
Right. Right.
But still, it is a system where you need to ask, where do we have to stop, yeah?
Yeah.
Because we don't want that this backfires.
Yeah. Yeah.
So we work with ethicists. We call this embedded ethics. So they're participating in our biweekly lab discussions. We discuss setups. They come to the lab, look into what we are doing, and we're also writing about this. Yeah, trying to find what have people said, which is meaningful, which resonates with our stuff.
Yeah.
So I'm really getting more and more into also such type of ethical, but also social science and political topics.
Mm-hmm.
Because you need to know what is politically on societal level acceptable. Yeah.
Yeah, for sure. So you're kind of entering into my area of interest and my area of expertise with these are mini brains that you're working with.
Yeah.
My issues with the development of kind of artificial life, I suppose we could call it that, is at what point do you think you would know such mini brains were sentient or conscious or self-aware or could feel pain or could suffer? Do you have any benchmarks, anything that might indicate that, or is that inherently part of the problem of doing this type of research that you might not know when?
I think it is a part of the challenge, and we have just published a paper with some colleagues in this field, and which is a call for clarifying the terminology,
Mm-hmm.
because we have to define what is consciousness in at the level of an organoid of a small thing. You have to understand: can you have pain without pain receptors? Um, but still, it could be unethical because there could be other types of suffering by such a system. And
Right.
we're discussing whether this is actually opening up for something you could call experimental ethics,
Yes.
because you can start verifying and measuring if you have a surrogate measure of any of these components. Yeah,
Mm-hmm.
define what is self-aware. Yeah, self-aware could be something as simple as where am I within my environment? Yeah, where I'm
Yes.
placed. That's already self-awareness. It's not a self-awareness which I would find critical,
Mm-hmm.
that I say, okay, just because this thing knows where it is in the culture plate, But we need to define it, yeah. And this is a very important first step. And then you can at some point look into how big does a brain need to be before you can start. It
Mm.
shows properties which you benchmark probably against different species. You say, okay, now it is on the level of a flatworm. Now it is at the level of an insect. Now it's on the level of a small mammal. Yeah,
Hmm.
and like this, you can also then set boundaries. Yeah, you might want to say, okay, we allow abortion up to a certain time.
Right.
Uh, if you say this is the function of a brain of an embryo at this time point, this could be a benchmark for us, yeah, to say,
Yes, right.
yeah, if you go any further. But I'm not saying this is the benchmark. I'm just saying you could start looking into such measures as an agreed thing we don't want to cross.
Yeah. Oh, yeah.
Um,
Very,
At the
very.
moment, we are interviewing lay people, just people, and ask them here, look, this is our research.
Mhm.
Um, when does it start to feel, "Ugh, perhaps you should better not?" But
Right.
also, how does it change if we tell you that we are trying to find medicines for Alzheimer doing this?
Mm.
So is it a communication issue, or is it really ethical principles where people say, no, not with my cells or not with my taxpayer money. This is not research which should take place. Yeah. So we're trying to find a starting point here. Yeah.
Oh, very good. Very good. Interesting. Now, when you were growing up, did you have role models? And if so, who are they?
Thomas Edison was one of them.
Edison. Interesting.
Yeah. I read several of biographies. I was so impressed with his um, enormous productivity and
Hmm.
work ethics.
Hmm.
More than 2,000 patents at the time and so many fundamental things.
Right.
I went into let's say engineering or any physics type of work. I'm yeah. I did mathematics, informatics sometime, but it's medicine and biochemistry which I did study.
Right.
But still, I was fascinated how a single individual can start so many things.
Interesting. My one of my heroes was Tesla. So
Yeah, very similar.
yeah, Tesla and Edison kind of
They were rivals. Yeah.
they were rivals for sure.
Yeah. With
For
my
sure.
love for Italy now, I would probably have chosen Tesla, but it was simply Edison which came first.
Well, I have Italian ethnicity, but I've never been, to Italy. But it, I'm dying to go there and to enjoy its culture and its people and everything else. But
I'm looking
um,
on the Lago Maggiore while we are talking, so it's a so I'm in Italy
you,
spending my summer here.
yeah, that's wonderful. That's wonderful. Now, are
And
you?
among us, I miss Italy more than I miss Germany, but don't tell anyone.
Oh, gee, my mother was German, and
Okay.
my father was Italian, and neither of them spoke the language in the house. So I'm sadly monolinguistic. But it was interesting growing up and enriched, by those two different cultures. Um, are you finding it all in your research as you move ahead further and further that um, colleagues are changing at all in their scientific research towards more humane treatments for lab animals? Are you seeing any kind of a shift, a paradigm shift in this regard? Is it catching on, like, say, more so in Japan or less in Portugal? Or like, are you seeing any differentiation worldwide for the work that you're doing and similar?
I mean, the first thing which we see is that society and scientific communities, they're all shifting slowly but surely towards abandoning animal testing or limiting it at least.
Right.
We have seen over the last 20 years in the US that uh, a gain of, let's say, opposition to animal testing for medical research
Hmm.
is about 10 to 15%. So that we once again at the moment about split half. but interesting, not along party lines. Yeah, there's a lot of Democrats in the US who are actually more leaning towards the animal testing to find problems with chemicals, to make the environment safer and protect lives.
Oh, interesting.
While there's interesting a lot of conservatives in who understand that animal suffering is not something they would like to support or see the business opportunities of new approaches. Yeah. Europe is on average 10% ahead. Yeah, so it's
Mhm.
about 60%. So the majority of Europeans don't want animal testing.
Right.
And politicians hear this. And the scientific communities also see that there is change. The
Mhm.
money goes more and more into these new approaches. It's simply more sexy. And also the animal experiments have to some extent been maxed out. So they have you cannot make them much different, and you cannot make them much more efficient. On the contrary, the better you make them, the more expensive they are,
Mm.
less competitive they are to other methodologies, also with respect to time.
Right.
If you want to know, for example, whether a chemical is producing cancer, this is something really people would like to know.
Sure.
This takes you 4 to 5 years to get the information
Hmm.
with the standard methodologies and does cost about 1 million. Yeah, 1 million.
Wow.
And yeah, so you can imagine how many substances are being tested with these type of methods. It's
Yeah.
limited, yeah.
Yeah.
Also because nobody wants to wait for results that long, yeah. And this is a value proposition of the new methods, that you can get information simply faster, and increasingly we're able to show also more relevant information with human cells instead of animals, yeah,
Mm.
and so on. So it is really, there's economic reasons also to substitute.
Yeah. So with culture, with cellular culture testing, are you using like human cells? Is that the idea? And testing on those versus using animal models?
I mean, it starts with the cells, but
Hmm.
we have also to see that cell culture is changing.
Hmm.
The cell culture of the past was very often looking like pan-fried eggs sunny side up in
Yep.
a petri dish. What we are moving to, and this is something my group is very much involved in, is to reproduce organs. So you have to see that a lung, for example, has more than 40 different cell types.
Wow.
So culturing a single of these is only giving us a very partial answer.
Right.
And we are trying to make these models more and more complete so that we replicate aspects of the architecture and of the function of the organ.
Mm.
And this is now called a microphysiological system. So micro is small, but it's physiological. So it's replicating function.
Right.
And these MPS, we call them microphysiological systems, is at the moment a disruptive technology. And we have been instrumental to create some really important opinion papers on this with larger groups of opinion leaders, but also we did carry out the 3 world summits in this field.
Mm.
We started in 2022 in New Orleans. We're in Berlin last year, this year in San Diego, in Seattle, sorry, it's in Seattle.
Yep.
Next year we'll be in Brussels, and then we hope to go to Asia. And my group has organized these. We had 1,300 people in Berlin, yeah, attending the conference.
Wow.
And we would have topped this in Seattle, but 400 Chinese didn't get visa in time.
Oh, yeah.
They're not allowed to come over. And but that's a different story. But
Mhm.
I'm just saying there is really a movement to these new methods,
Mhm.
and the regulators love them. The FDA, the Food and Drug Administration, which is the most important regulator in the world,
Yep.
they have reached out to us because of this, and we signed a research collaboration agreement with them earlier this year.
Mhm.
And this research collaboration agreement is to help building trust in these methods. It's to educate FDA staff and give these activities visibility to the outside. Yeah.
Mhm.
So, I mean, you have to imagine a 19,000 people agency knocks on our doors and says, you guys here at Hopkins, you can help us. This is a dream come true.
Yes. Oh, very good. Now, have you faced any pushback from dissenters in other fields of science or colleagues to say, no, 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 I'm always going to use animal models. I think what you're doing is just, you're going in the wrong direction. Has there been any pushback at all?
Yes, sure. I mean, there's
Yeah.
people who are diehards in the sense, yeah. They want, this is our daily experience, that there is people who we have to convince, yeah.
Hmm.
Very often this is not easy because I can reach out very easily and preach to the converted because we go, if we stay in our bubble,
Yep.
I need the challenge of other groups, yeah, and
Of course.
I need arguments, lines of evidence to really um, pull them, and this is to pull them on to our side. And this is really not the ethical discussion. This is very often the economical discussion. It's about reproducibility of science. It is about the cost, how fast you can produce scientific knowledge, at which price. Yeah.
Yeah. Yeah. That's it. If you had a kind of a magic wand and you could create something that's just in your mind for lab testing and the future of toxicology and that sort of thing, how would AI generate that? What is missing now that you hope AI is going to be able to produce in the next, say, 4 to 5 years?
I mean, we're trying at the moment to do something which is crazy.
Hmm.
We want a co-pilot, we call it, for
Hmm.
toxicology. We want to do something where you put in a structure,
Hmm.
and then the AI is crunching the numbers and looking into all what we know about chemicals which are similar, what we have known about what the data we already have on a substance, how they would predict what they would suggest with which probability for the overall outcome. Let's say we don't have done a cancer test because this was 1 million, but we have already a few other informations. Sometimes also things like, is it a soluble substance? Is it a substance which is more lipid-like, or is it more water-like,
Hmm.
hydrophobe or hydrophobic? This is a very important piece of information. It's not difficult to either measure or predict, but it is an extremely valuable piece of information because it also already tells you whether it can accumulate in humans. Yeah?
Hmm.
And this co-pilot will probably be terrible. It will not often be correct, but we can then start looking into what information would make it better. Yeah,
Yeah.
where does this uncertainty come from? And then we can start engineer it. And I'm so far we are blown away with the test results we have so far, but I believe it when we have really had it tested, externally evaluated. But we're coming closer, yeah.
Good.
And then we will, this is, if you want to change from one technology to the other,
Hmm.
it is normally not changing today to something different.
Right.
When we moved from horse coaches to cars, it was not that it one day after the Model T came out, everybody said, okay, let's go from horse coach to cars. You need infrastructure, you need experience, you need to optimize these products.
Hmm.
You need to know what they can and what they can't, yeah.
Yeah.
And then at some point, it will be ridiculous to stay with horse coaches,
Yeah.
and or they have uses which are romantic or museum-like, yeah.
Hmm.
And I hope that we reach something like this where we simply make a value proposition with this new method that people increasingly use them.
Yeah.
Yeah? And then you can suddenly also use them in a completely different way. For example, in toxicology again, if you work on structure only, then the chemist can already check out before even synthesizing a chemical whether it makes sense, and then say 80% likelihood that this is producing allergies is enough for me to go for something which has 10%
Mm.
likelihood. Yeah. We call this green toxicology, so that in the end, much safer chemicals are being produced that in product development, we choose safer substances, so we have less surprises at late stages. After a lot of money has been spent, and where typically management is not very eager to start from scratch. Yeah.
Yeah. Yeah. Very good. Very good. Final question. What's next on your to-do list?
Next thing is actually something big.
Yeah.
Because I'm not shying away from big things, as you
Good. I like it. I like it.
You know, over the last 40 years, the driving force in biomedical research has been the Human Genome Project,
That's right.
which has really given us a completely different angle on how to understand disease, how to test, to develop new medicines. But after 40 years, we know that 5% of diseases are purely genetic, and 40% roughly have a genetic component, which is important. But this is still exposure of some kind for 70 to 80% of chemicals. And what we want is now, after the Human Genome Project, the Human Exposome Project.
The Human
Exposome Project, which is cataloging exposure contribution to disease.
Wow.
And what I hope is really that we move from the art of poisoning rats to what is the exposure contribution to disease.
Interesting.
So I would hope that essentially we dissolve toxicology as the naysayers, which are just saying this cannot proceed to the market, or so, to something which is a meaningful contribution for all of human disease. And I'm not alone. There's many people. The concept of the human exposome is from 2007. Yeah. So interestingly, by the head of the Cancer Research Institute, IARC, the International Institute in Lyon. And we have now forming an alliance. And next year in May, we got from Hopkins a fantastic venue, only a few hundred meters from the Capitol in Washington, to form a museum, and we have this for free to bring together stakeholders. And at the moment, we are trying to get these opinion leaders from all over the world together, agencies, to form a public-private partnership in order to move to human exposome. And we call it the human exposome moonshot because it's a moonshot. It's reaching very, very far.
Yeah.
Might not reach the moon, but already getting into the sky is something.
No, I love it. I love big ideas and big projects. Somebody has to do them, so why not? Why not you? Right?
No, we have very, very powerful partners, but we use the flexibility of a university group, the fact that we're not in an agency with red tape, to get these people together. Yeah. So we teamed up instantly with the National Institute for Environmental Health Sciences. Who are excited about this, and they are happily working with us. The director, Rick Wojcik, will be our opening keynote lecturer, and his team is part of the organizing committee now.
Oh, good.
Because they love that outside support can communicate in a fast and informal way with people where you otherwise have to go through government relations and so on.
That's right. Listen, sir, it's been a pleasure talking to you and finding out more about your research and the use of AI and toxicology research. This has been fascinating. And I want to thank you for participating today. Thank you very much.
Thanks for having me.
Thank you.